Hello students, welcome again to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor at IIT Delhi. Today we are going to talk on the module called Myeloid System from the paper Immunology. This is the first part of our discussion on this, and we will continue discussing it in a subsequent module as well. In today's lecture, we will learn that various blood cells arise from a multipotential stem cells. B and T lymphocytes mature in the bone marrow and thymus respectively. After this, they move to or circulate among secondary lymphoid organs scattered throughout the body. At the moment, we will use the term stem cell in the generic terms. Much later, almost towards the end of this paper, we are going to be discussing stem cells in more details, and there we will actually talk of the finer nuances in the meaning of the word or the concept stem cell. The objectives of this particular module are to learn how stem cells first differentiates into lymphoid and myeloid progenitors. After that, we will learn that how B and T lymphocytes, the two classes of lymphocytes, both mature in their respective primary lymphoid organs. Then we will learn that after this, how these lymphocytes circulate throughout the body, and then the immune response is initiated in the secondary lymphoid organs. So the concept involved basically is the formation of the cells of the blood. This process is called hematopoiesis, while lymphocytes are called lymphoid cells. Other blood cells have a myeloid progenitor cell. So in the beginning, the fertilized egg has a multipotential stem cell. This multipotential stem cell can develop into other kinds of stem cells, but this multipotential stem cells is the one from where all the cells of the body of the animal ultimately are formed by processes of differentiations. This multipotential stem cell, hence in our present context, differentiates into lymphoid cell progenitor and myeloid cell progenitor. Bone marrow and thymus are primary lymphoid organs. On the other hand, the spleen, lymph nodes and mucosa associated lymphoid tissues are secondary lymphoid organs. Let us look at the multipotential hematopoietic stem cells. This is the stem cell which, upon differentiation, gives rise to all the cells which are present in the blood. The development of these blood cells is called hematopoiesis, and that is why this stem cell is called hematopoietic stem cell. The phenomenon of the hematopoiesis begins in the early yolk sac. During embryogenesis, this process moves over to the fetal liver, fetal spleen, and in the bone marrow of the neonate and the adult. Hence, the common multipotential hematopoietic stem cells are found in the fetal liver, fetal spleen, and in the bone marrow. It is in the bone marrow that hemopoiesis continues throughout the life. It is how these cells replicate and seed other organs during the life of the adult. As this slide traces the hemopoiesis, initially the blood stem cells forms the myeloid stem cells and the lymphoid stem cells. It is the myeloid stem cells which then gives rise to the various cells like red blood cells. The lymphoid stem cells, on the other hand, gives rise to the white blood cells, which include not only the B lymphocytes and the T lymphocytes, which mediate the cellular immunity. Also, the natural killer cells also arise from this lymphoid stem cell. The differentiation of the stem cell is dependent upon its microenvironments, that is, the nature of the organ or the tissue. This differentiation involves stromal cells and specific cytokines. Within the fetal liver, thymus, and bone marrow, different stromal cells. These stromal cells include macrophages, endothelial cells, epithelial cells, fibroblasts, and adipocytes. So these different stromal cells creates foci. At different foci, different cells can develop. Both cytokines and adhesion molecules play important roles. It is these interactions which switch on gene expression, which is necessary for the functioning of that particular differentiated cells. The various interactions which occur during this process are also shown in this figure. This is a very important slide because this shows the different kind of a cells which we'll be talking about throughout this discussion on immunology. This shows the detailed outline of hematopoiesis. We have already mentioned that the all blood cells arise from a common stem cell. We also mentioned that this pluripotent hematopoietic stem cells gives rise to initially two different types of stem cells: common lymphoid progenitor, which gives rise to lymphocytes; common myeloid progenitor, which gives rise to granulocytes, which are also called polymorph nuclear white blood cells or leukocytes. The red blood cells and platelets also arise from this common myeloid progenitor. The differentiated myeloid cells, which occur further down the line, and are present in the different tissues are mast cells, macrophages, and immature dendritic cells. Lymphoid organs contain lymphocytes in the framework of lymphoid cells. The interaction of lymphocytes with the non-lymphocyte cells is responsible for lymphocyte development, initiation of the adaptive immune response, maintenance of the lymphocytes. Lymphoid organs basically are of two kinds: central, or what we refer to as primary lymphoid organs. The second kind, which we refer to as secondary lymphoid organs, are also called peripheral lymphoid organs. Lymphocytes are generated in the primary lymphoid organs. So, adaptive immune response is initiated in the secondary lymphoid organs. It is here that the lymphocytes show the sustained action. There are two kinds of primary lymphoid organs: bone marrow and thymus. The bone marrow is an important hematopoietic tissue present in the long bones and axial skeleton. Venous sinuses are around a central artery and vein and enmeshed in developing cells. All blood cells are derived from the bone marrow stem cells. About 10% of the bone marrow cells are lymphocytes. This goes on to emphasize the important role of the bone marrow in production of the cells which are involved in the immune response. As we said, B cells differentiate further in the fetal liver and bone marrow. B lymphocyte precursors are present in the fetal liver by 8 to 9 weeks of pregnancy in humans. This period is about 14 days in mouse. In later life, bone marrow takes over the production of B lymphocytes. Both B and T lymphocytes are formed in the bone marrow. B lymphocytes develop in the bone marrow itself. T cells, on the other hand, migrate to the thymus and develop there. There is an organ called bursa fabriceus. At one time, it was believed that this organ is the site of the B cell development. Contrary to what some books claim, B cells were so named because of this belief. It is fortuitous that the bone marrow also has B as its first letter. Bursa fabriceus is present in the common exit of intestinal and genitourinary tract in birds. It has been shown that the bursectomy of young chickens exhibits antibody synthesis. Bursa fabriceus is the site where B cells differentiate in birds. It atrophies with age. Let us look at the thymus. This organ is very important in the context of the immune response, particularly the cell-mediated immunity. In mammals, it's a bilobed organ and located in the thorax. Each lobe has lobules separated by connective tissue. Each lobule has inner medulla containing mature T cells and outer cortex which is more tightly packed with immature and proliferating T cells. Many epithelial cells present in the lobules play an important role in differentiation. In mammals, atrophy of thymus begins with puberty and its decline continues with age. Pregnancy and stress both are known to promote thymus atrophy. Let us now look at the secondary lymphoid organs. It is here in these secondary lymphoid organs that the immune response actually takes place. Spleen is the first important secondary lymphoid organ. Then comes lymph nodes. Mucosa associated lymphoid tissues are called MALT and they are shown in this figure how they are distributed. Once the B and T cells have matured, they migrate to these secondary lymphoid tissues. This shows the circulation of various cells. The cells which are present in the blood, of course, and the antibody molecules which are present in the blood. They circulate through tissues via the bloodstream. The lymphatic vessels return these cells and antibodies from the tissues to the bloodstream. The circulation of the lymphocytes between the tissues, the blood and the lymph glands then encounter with antigen or enable their participation where the immune response to the antigen has been already initiated. Spleen is another important secondary immune organ. The spleen is an encapsulated organ. Its interior splenic pulp is spongy in appearance. As we can see here, there is a white pulp and there is a red pulp. The white pulp islands are situated within the red pulp, which is a network of red blood cells, macrophages, and the B cells. The spleen traps the microbes which enter it via the bloodstream. It also has another important function, that it removes the dead red blood cells and also the immune complexes. Individuals with the spleen removed, that is splectnomized, are at a high risk to infections from encapsulated bacteria and malaria, and that shows the important role which spleen plays, more so in case of immune response against certain infections. We mentioned lymph nodes. The distribution of lymph nodes in the body is shown in this figure. Lymph nodes are actually small spherical encapsulated structures. Their size normally is in the range of 2 to 10 nanometers. As can be seen here, they are located at various parts of the lymphatic system. They are large groups of lymph nodes at specific places, which are axillae, groin, and neck. Lymph nodes essentially trap antigens from the circulating lymph. It is the circulating lymph within the tissue which brings the antigens to the tissues, and lymph nodes trap these antigens there. We also mentioned earlier mucosa associated lymphoid tissues, which is abbreviated as MALT. Here in this figure, lymphoid tissue from mucosa of small intestine is shown, magnified 14 times. Mucosa associated lymphoid tissues is an uncapsulated lymphoid organ. Greater than 50% of the lymphoid tissue in human body is a part of MALT, and that shows the importance of the malt in the overall scheme of immune responses. There are actually three tracts which are covered by these mucosal surfaces. These are also the main sites for the entry of microbes. The mucosal surfaces then are associated with respiratory, digestive, and genitourinary tracts. Malt tissues are of two kinds. One class is those of subepithelial lymphocytes forming follicles covered with specialized epithelial cells called microfold or M cells. These M cells are shown there in the figure. The second kind of malt tissue cells are essentially diffused cell populations below the epithelial cell surfaces. Depending upon their association with a specific kind of tissue, malt can be subdivided into knalt, balt, and galt. Knalt stands for nasal associated lymphoid tissues. BALT stands for bronchus associated lymphoid tissues. GALT stands for gut associated lymphoid tissues. Hence, MALT is associated with different tissues. Some immunologists believe that these different types of the malt, that is the NALT, BALT, and GALT, may be forming an interconnected system. Let us look at the NALT first. NALT consists of lymphoid tissue at the back of the nose, that is the pharyngeal, tonsil, and other tissues, and also the one associated with what are called Welde's rings. These are present in palatine and lingual tonsils, and this is the lymphoid tissue of the neck and pharynx. NALT essentially handles airborne microorganisms. These tissues, unlike lymphatic nodes, do not have lymphatics. Tonsillar lymphoid tissue, for example, there the antigens trapped in the crypts are transported by M cells into the subepithelial areas where the lymphocytes are stimulated via the antigen presenting cells. The second class of malt which we mentioned was of course BALT. BALT consists of aggregates of lymphocytes organized into follicles present in all lobes of the lung. These are shown here in this figure in detail. These are situated under the epithelium, mostly along the bronchi. M cells, which we mentioned earlier, they transport trapped antigens to the underlying cells, which include lymphocytes. Then we have GALT. Microbes trying to enter the body via the intestinal tract are prevented by GALT. GALT hence has to have a mechanism which distinguishes between harmless food and infecting microbes. This mechanism consists of microfolds, M cells on the epithelia, and associated antigen presenting cells, APCs. So, hence a complex mechanism involving these APCs, and the T cells decides whether the sample antigen results in tolerance, that means no immune response is initiated, or should it provoke an immune response. In fact, Similar mechanisms also exist in bald, gnarled, and lymphoid tissues, which are associated with genitourinary tracts. Pairs patches are aggregations of the lymphoid tissue which belong to galled. In humans, it is commonly found in the lowest portion of the small intestine, that is ileum. Macroscopically, Pairs patches appears as oval or round lymphoid follicles. Pairs patches uptake of largest particles is allowed by M cells that are found in the follicle associated epithelium. M cells have the unique ability to sample an antigen from the lumen of the small intestine and deliver it via transcystosis to antigen presenting cells and lymphocytes located on their basolateral side. The Pairs patches, as we mentioned, are aggregates of lymphocytes which appear as pale patch on the gut wall. So, students, let's now summarize what we have learned in this module. So, we learned that the differentiation of the multipotential cell gives rise to lymphocytes. B lymphocytes and T lymphocytes mature in the bone marrow and thymus respectively. Lymphocytes circulate in the tissues via the lymphatic circulation and reach there via the blood circulation. The secondary lymphoid organs, which are spleen, lymph nodes, and malt, they help in trapping of the antigen and then its processing. We have also seen that the differentiation of the multipotential stem cells gives rise to two main kinds of cells: cells of the lymphoid system and cells of the myeloid system. So we have also discussed the lymphocytes in this lecture. Thank you.